The overall goal of this procedure is to assess membrane repair capacity in both the animal and isolated cells. Isolated skeletal muscle fibers are collected and surrounded with media containing FM143 dye. A UV laser is then used to irradiate and damage a portion of the cell membrane to allow entry of the dye into the cell. Isolated cells are transfected with plasmid DNA expressing GFP MG53, which will localize to the plasma membrane and cytosol of the cell. The tip of a microelectrode is then inserted into the C2-C12 membrane to induce damage. Ultimately, results can be obtained that show translocation of the GFP MG53 to the injury site.
Visual demonstration of this method is critical since practice is required to master the techniques of microelectrode penetration and muscle fiber isolation.
Demonstrating the procedure will be Dr. Peihui, our lab manager, Dr. Hua Zhu, a postdoctoral fellow, Matt Orange, a graduate student, and Dr. Xiaoli Zhao, a research assistant professor in our department.
To begin, place a tray or a blue lab pad underneath the treadmill to collect waste from the animals during the experiment. Next, set the angle of the treadmill surface. Some treadmills have an integral apparatus to adjust the incline, while others require the treadmill to be elevated through other means. A flat surface or an angle between 7 and 15 degrees downhill or uphill is generally used. Acclimate the mice by placing them onto the treadmill with the electric grid off and the belt drive motor.